Our next speaker is Robert Kelly. Robert is group leader at the Institute of Developmental Biology at the University of Marseille, AixMarseilles. Robert is a developmental biologist whose major research interest is in the secondary heart field, and more specifically, the developmental potential of this progenitor population and its relevance for congenital heart disease, including and especially that associated with 22q11 Deletion syndrome. Robert, we can see your presentation. Thank you very much.
Great. Thank you, Elizabeth. And I'd like to thank the organizers for the opportunity to join this exciting meeting today and tell you about our work in the mouse on TBX1 function in the second heart field and cardiopharyngeal mesoderm. So the heart starts out in the early embryo as a transiently linear tubular structure, which we can see here, situated under the head folds. The blood enters through the venous pole, situated posteriorly, and exits through the arterial pole, anteriorly. And this early heart tube is derived from a population of progenitor cells called the first heart field, and then rapidly extends in length and loops to the right by addition of progenitor cells called from the second heart field, a progenitor population situated in mesoderm lateral to and underlying the pharynx, so pharyngeal mesoderm, which in this transverse section of an embryo at this time point, we can see forms an epithelium in the dorsal pericardial wall behind the heart. And it's from this position that these cells add on to the rapidly growing arterial and venous poles of the heart, such that halfway through mouse gestation, approximately equivalent to the 5th week of human development, we can see the embryonic heart in which the second heart field has given rise to the right ventricle, outflow tract, and a large part of the atrial myocardium. And this phase of heart tube elongation is followed by the phase of cardiac septation and chamber growth. We can see the formation of a ventricular septa, atrial septum has formed, and this myocardial cylinder, that's the embryonic outflow tract, has divided to generate the ascending aorta and pulmonary trunk, the outlets of the definitive right and left ventricle. Now, defects in this septation process or in the prior addition of progenitor cells from the second heart field during heart tube elongation, which creates the template on which septation takes place, result in a spectrum of congenital heart anomalies. And here's the example of tetralogy of Fallot, where we can see that the ascending aorta has failed to make the critical exclusive connection with the left ventricle. And what I'd like to do today in this talk is firstly discuss patterning of the second heart field, and we'll be looking at how progenitor cells add on up to the arterial pole versus the venous pole. And then in the second part of the talk, I'm going to position this progenitor cell population in the context of a larger mesodermal progenitor cell population in the pharyngeal region that give rise not only to the heart, but also to skeletal muscles of the head and neck. And in particular, we'll be seeing that they also give rise to connective tissue of certain neck muscles. And in each of these processes, I'll be pointing out the function of TBX1, the 22q gene. So before I get to patterning the second heart field, I'd just like to say that the first and second heart field arise very early in development at the time of gastrulation and independently activate the first cardiac transcription factor, MESP1. And this very early embryological origin is reflected by an early evolutionary origin. If we look at the single ventricle of the zebrafish, we can see that the fish ventricle has a second heart field component and a first heart field component. And this tells us that the difference between the single fish ventricle and our two ventricles isn't that we've added a progenitor cell population that's given rise to an entire chamber, but that a septum has formed at the interface between the contribution of these two progenitor cell lineages in the middle of the ventricle. And this is actually what happens during mammalian development as well. And this also happens during atrial development. Here we can see in this cartoon, second heart field cells under stimuli from pharyngeal, from pulmonary endoderm, entering the embryonic atrium, dividing it into future right and left atrial chambers. So the second heart field is patterned into distinct anterior and posterior domains. We can see here TBX1 expression expressed in the anterior part of this epithelium. Here we're looking in a sagittal section in the same plane as this view here. And we can see that within this epithelium, there's a very sharp boundary. And in the posterior part, we can see that TBX5 is. And This anterior domain, labeled by TBX1, contributes to the arterial pole of the heart. Defects in anterior second heart field development result in outflow tract or conotruncal congenital heart defects that we've already heard about, including double outlet right ventricle. Here again, you can see tetralogy of Fallot or ventricular septal defects. Whereas defects in the TBX5 positive posterior second heart field cause venous pole abnormalities, including atrial septal defects and atrioventricular septal defects. And of course, TBX1 and TBX5 are major genes for human genetic syndromes with cardiac defects. And here we can see the common arterial trunk that we've already seen today in a TBX1 homozygous mutant mouse embryo. And here in a TBX5 haploinsufficient embryo, we can see an atrial septal defect. And TBX5, of course, being the major gene for Holt-Oram syndrome. So we set out a number of years ago to ask how this sharp boundary between TBX1 and TBX5 expression forms in this epithelium. And we looked at the expression pattern of these two transcription factors at different time points during heart development. At early stages, TBX5 is expressed in the first heart field and the earliest differentiating cardiomyocytes, but not in TBX1 positive pharyngeal mesoderm. As the heart tube forms, we start to activate TBX5 in TBX1 positive pharyngeal mesoderm close to the venous pole of the heart. And we can see here these yellow nuclei, which are co-expressing both of these transcription factors. 24 hours later, this region of overlap is resolved to form the very sharp boundary that I showed you earlier. And we know that this occurs by downregulation of TBX1 in these co-expressing progenitor cells, because genetic lineage labeling from the Bruno lab has shown that TBX5 expression only occurs in cells that contribute to the venous pole of the heart. We can see this downregulation of transcription in the posterior second heart field with another marker. This is an enhancer driving CRE in a transgene, and this enhancer is from the MEF2C transcription factor locus. And if we look at CRE expression in this epithelium behind the heart, we can see that just like TBX1, it's expressed in the anterior part, but not the TBX5 positive posterior part. However, if we look at the genetic lineage driven by this enhancer, we can see that this does contribute to the posterior part, and thus, like TBX1, must be downregulated after TBX5 is activated. And indeed, this downregulation requires TBX1. In a TBX1 mutant embryo, we can see that the CRE expression continues right to the venous pole of the heart. This posterior part of the second heart field makes a very important contribution to the heart. It gives rise to atrial septal structures. We're looking here in a four-chambered view of the fetal heart. We're using the same MEF2C enhancer CRE transgene, this time driving expression of a conditional beta-galactosidase reporter gene. We can see here expression of beta-galactosidase in cells derived that have added on at the arterial pole of the heart and the right ventricle, the right side of the interventricular septum. And we can see at the venous pole contribution to atrial septal structures, the primary atrial septum, and this population of cells here called the dorsal mesenchymal protrusion that makes an important bridge between the primary atrial septum and the atrioventricular septal complex. And in TBX1 homozygous mutant embryos, where the patterning of this posterior part of the second heart field is perturbed, we can see that the DMP, this dorsal mesenchymal protrusion, fails to contribute to the heart. And we observe this defect in about 30% of TBX1 homozygous mutant fetuses. And this is very similar to the atrial septal defects that are seen in TBX5 haploinsufficient embryos, as we saw earlier. Now, this boundary, so this takes us to a model where we have a second heart field program where TBX1 and this MEF2C enhancer are expressed. And this is maintained in cells that will contribute to the arterial pole of the heart. However, in the posterior part of the second heart field, TBX5 is upregulated. There's transient co-expression followed by downregulation of the anterior program and specification of these venous pole progenitor cells in the posterior second heart field. We wanted to ask how this activation of TBX5 takes place. Through experiments that I don't have time to go into, we know that this is de novo upregulation of TBX5. And we knew that retinoic acid signaling components, including the biosynthetic enzyme RALDH2, is expressed in mesoderm immediately adjacent to the posterior second heart field and overlapping with this region. And by blocking retinoic acid signaling using an antagonist that's a pan blocker of retinoic acid receptor activity, we can fail. We lead this blocking this at prior to the upregulation of TBX5 leads to failure of TBX5 activation in cells around the venous pole of the heart. Here we can see TBX5 in red. We're looking ventrally onto this epithelium of progenitor cells, and we can see that this upregulation fails if we expose the embryos to this antagonist. And these embryos will go on to develop hearts which lack the dorsal mesenchymal protrusion, very similar to the phenotype I've showed you in TBX1 null embryos or TBX5 haploinsufficient embryos. Whereas, and if we block retinoic acid signaling after upregulation of TBX5, we no longer see this defect. We can see that the DMP forms. These hearts do have outflow tract defects because retinoic acid signaling is also required for outflow tract defect. But the inflow tract defects, this atrial septal defect, requires retinoic acid antagonism prior to TBX5 activation. Now, this boundary between TBX1 and TBX5 that defines the sites of future atrial septal structures is also seen at the sites of interventricular septal formation. Here we're looking at the comparison of a TBX1 Cree lineage and a TBX5 Cree lineage, and you can see that they meet at the site of the interventricular septum. And this similarity extends to analysis of the MEF2C transgene. We can see here that the transgene driving beta-galactosidase expression is observed in the right ventricle and outflow tract, but the genetic lineage driven by this enhancer contributes also to the left ventricle where TBX5 is expressed. And this tells us that when TBX5 is expressed, activation of the enhancer driving this from this MEF2C locus is repressed. And this is extremely similar to what I've just shown you is taking place at the site of atrial septal formation here in the posterior second heart field. And this leads us to the concept that this transient overlap between the second heart field pharyngeal mesodermal gene expression program and the first heart field TBX5 program may be very important in defining boundary formation, both at the sites of atrial septal formation and at the sites of ventricular septal formation. And mining this transient co-expressing population may give us very important information for understanding the origins of septal defects, both atrial and ventricular septal defects together, accounting for over 50% of congenital heart defects. Now, in the second part of the talk, I'd like to show you that the second heart field originates in a population of mesoderm called cardiopharyngeal mesoderm that also gives rise to craniofacial and neck muscles. And these muscles originate from the mesodermal core of the 5 bilateral pharyngeal arches that form either side of the pharynx. And muscles at mesoderm in the first arch gives rise to the muscles that open and close the jaw. And clonal analysis experiments that I don't have time to go into in this short talk have shown that these cells are clonally related to myocardium in the right ventricle of the heart. Muscles from the second arch, muscles of facial expression, are clonally related to myocardium at the outflow tract of the heart, and muscles derived from the posterior arches, neck muscles, are clonally related to myocardium in the inflow tract, atrial myocardium, as well as a small population of outflow tract myocardium. And this common origin of particular muscles and particular parts of the heart is very important if we want to understand genetic defects that affect craniofacial and cardiovascular development, such as those seen in 22q patients. And how can it be then that cells in the muscles that open and close our jaw are clonally related to cells in the right ventricle? Well, the answer is that the early heart forms in front of the face in the very early embryo, when it's a linear first heart field derived structure. Then the first pharyngeal arch forms, the arches form in sequence, mesoderm in this region contributes right ventricular cells to the heart and the progenitors of first arch muscles. Then the heart progressively connects to the second arch as this forms. The outflow tract starts to form and second arch muscles form and so on in a posterior direction. And we thus have 3 bipotent populations of progenitor cells in the pharyngeal region that give rise to specific head muscles in different parts of the heart. And these are very closely related to the 3 streams of cranial and neural crest cells that move through the pharyngeal region, some of which also contribute to the heart, as we've heard. And coordination between neural crest cells and cardiopharyngeal mesoderm plays a critical role in orchestrating pharyngeal development, craniofacial and cardiovascular development as well. Now, these muscles that are derived from cardiopharyngeal mesoderm are TBX1 dependent. We showed a number of years ago that here we're looking at the expression pattern of the myogenic regulator MyoD, and we can see that in a TBX1 null embryo here at the bottom, the myotomal muscles derived from the somites and the Semitic muscles in the forelimb, and this population here, the hypoglossal cord. This is a somite-derived population of cells that will give rise to the tongue muscle and ventral neck muscles. These are all present in a TBX1 null embryo, but the sites of branchiomeric myogenesis here in the pharyngeal arches fail. If we look at a mouse fetus at later stages of development, here we're looking under the mandible with another marker of the myogenic lineage. We can see muscles of facial expression, muscles that operate the pharynx and the larynx, and these are missing in the TBX1 null. We can see these four muscles radiating out at the base of the tongue. These are derived from the hypoglossal cord, a Semitic derivative. And we can compare this to sequential dissection of a human cadaver, where we remove the ventral neck muscles and we have these four muscles radiating out at the base of the tongue. And this very nicely shows us that TBX1 provides a genetic dissection and defines a population of TBX1 dependent branchiomeric skeletal muscles. And these extend to include esophageal skeletal muscle, which we can see here fails to activate, to differentiate in a TBX1 mutant embryo. And in fact, esophageal skeletal muscle forms from a population of cardiopharyngeal mesoderm that migrates down the esophagus and differentiates on a smooth muscle scaffold in a TBX1 dependent way. Now, what I'd like to do very briefly is show you that cardiopharyngeal mesoderm gives rise not only to the heart and skeletal muscle, but also to connective tissue of neck muscles. And here we're focusing on muscles in the laryngeal region. And we came across this when we were analyzing the precise contribution of this cardiopharyngeal mesodermal lineage to this region of the embryo. And we can see here that cricothyroid laryngeal muscle that's labeled by this genetic lineage. Here we're now using a fluorescent reporter gene, and we're labeling the muscle fibers in red. We can see that this adjacent muscle, the omohyoid, the fibers are not labeled by the genetic lineage, but they're positive for mice and heavy chain. But we can see that the lineage contributes to cells interspersed between the muscle fibers. And again, here in this other view of the omohyoid from down here, we can see that again, this interspersion between lineage negative fibers and also to the tendon region. And we suspected that this might be connective tissue, and looking with a transcription factor marker of connective tissue, TCF4, we can see in a branchiomeric muscle where both the muscle fibers and these interspersed cells are labeled by the lineage. We can see that these are co-localized with TCF4. And this is very clear here where we're looking at a Semitic muscle, again, the omohyoid, where we can see that fibers are lineage negative, but that the TCF positive cells between the muscle fibers are labeled by the GFP. Now, connective tissue plays a very important role in muscle organization and in muscle attachment sites. And here we're looking again at two laryngeal muscles, the posterior cricoarytenoid and the cricothyroid. And we can see in these two muscles, we have a very dense region of the muscle where both the fibers and the connective tissue are labeled, and another region of the muscle where only the fibers, but not the connective tissue, are labeled. And looking with another genetic lineage marker, this time tracing neural crest cell contributions to these muscles, we can see that neural crest cells actually contribute to the connective tissue in the region of these muscles where the connective tissue is not derived from the cardiopharyngeal mesodermal lineage. So here we have two types, two embryological origins of connective tissue in this muscle down different ends of the muscle. And it's not clear yet to what extent these converge on a common connective tissue phenotype, but we think that this is likely to be important in patterning these muscles and making sure that they make the right attachments at different sites of the muscle. And in order to investigate if cardiopharyngeal mesodermal derived connective tissue is important for muscle patterning, we turn to a master regulator of cardiopharyngeal development, which is TBX1. We asked if in a TBX1 mutant where all these muscles form, are any of them mispatterned in a heterozygous TBX1 mutant? And indeed, we observed muscle patterning defects in a TBX1 heterozygous mutant embryo. Here we can see the sternocleidomastoid muscle. This is a branchiomeric muscle, so derived from cardiopharyngeal mesoderm. We can see an abnormal attachment to this muscle to the greater horn of the hyoid bone in a TBX1 haploinsufficient fetus. And like other aspects of the TBX1 heterozygous phenotype, we observe this, including, for example, the fourth arch artery patterning defects. We can see that these defects can be unilateral or bilateral. We can see such defects also with muscles which are not branchiomeric, like the omohyoid. Here again, we can see a patterning defect where the omohyoid shows an abnormal high attachment to the greater horn of the hyoid bone. And we also see such defects in embryos where we've removed one copy of TBX1 exclusively in this cardiopharyngeal lineage using MEF2C cream. So what this tells us is that cardiopharyngeal mesoderm contributes myogenic progenitor cells, makes a cellular contribution to the muscle fibers themselves in these branchiomeric neck muscles, but it also makes a cellular contribution to connective tissue, both in branchiomeric and somite-derived neck muscles, and that this appears to be important for the patterning. And this raises interesting questions about when these different lineages diverge in the cardiopharyngeal mesodermal lineage and what the role of TBX1 might be in generating these different fates. And you'll recall that this mesoderm also gives rise to cardiomyocytes. So when these different branch points occur, remains to be discovered. I've also shown you that neural crest cells contribute connective tissue to both branchiomeric and somite-derived neck muscles, again illustrating the coordination of the contribution of these two embryonic cell populations to development of structures in the pharyngeal region. And indeed, understanding this coordination, as has been pointed out by Bernice and others, is likely to be key in understanding TBX1 haploinsufficient phenotypes. So just to summarize what I've shown you in this talk, we've seen in the first part that overlapping TBX1 and retinoic acid-dependent TBX5 expression establish a boundary within this progenitor cell epithelium and define future atrial septal structures that both atrial and ventricular septa form at the interface between heart fields. And then in the second part, I showed you that the second heart field derived parts of the heart and TBX1 dependent head muscles are derived from cardiopharyngeal mesoderm, and that cardiopharyngeal mesoderm also gives rise to connective tissue, and that this is important for patterning these both branchiomeric and non-branchiomeric somite-derived muscles, and that through unknown mechanisms, this is dependent on maximal TBX1 expression levels. And finally, I'd like to thank my colleagues in the group in Marseille who've contributed to this work. The work on the heart was initiated by Magali Thévenu-Ricci together with Christopher de Bono, and is now being continued by Clara Guijara-Calvo and Charlotte Tellier in the lab. And the work on skeletal muscle that I showed you is the work of Noritaka Adachi together with Camille Dumas. I'd like to thank our collaborators in Marseille and within the Leduc 22q network, Antonio Baldini and Bernice Morrow, and I'd like to thank you all for your attention.
Thank you very much, Robert, for a beautiful presentation, wonderful images, and also for reminding us very dramatically that 22q11 deletion syndrome is a multi-system disorder. So thank you very much, Robert.